Morning, shall we get started? It's just gone 10 o'clock, so we're slightly late in starting. So, my name is Munir Mohammed. I'm the chair of the UK Pharmacogenetic Stratified Medicine Network. Now, you probably all know what Genomics England is, but you probably don't know much about the UK Pharmacogenetic Stratified Medicine Network. And this is an organization which I formed about 10 years ago, largely to bring the community together in terms of the areas of pharmacogenomics and stratified medicine, really so that you could have stakeholders from many different organizations, academia, healthcare, the regulators and industry, trying to come together, networking and developing collaborations. And the network has been very successful. We fund ourselves and we run an annual meeting. Our next annual meeting is at the Royal College of Physicians on the 10th of March. Next year, and our annual meeting is usually attended by 250 people. We have to limit it to 250 because the Royal College Lecture Theatre can only take 250. And we've got John Bell opening our meeting next March, and a fantastic lineup. So I do encourage you to come to that. It's pretty cheap, and we are free to join as well. Okay, so free to join. You can join the UK Pharmacogenetic Stratified Medicine Network, and we're very nice. We'll give you cups of teas and things like that, and so on. So please do join us. So the idea, and we also hold workshops with other organisations. So we've held workshops with NHS England, Academy of Medical Sciences, with MHRA, and today, very grateful for Genomics England to be working with us. And I discussed with Ellie and with Mark Caulfield about a need for a workshop in this area, and I think it was Ellie, your suggestion about human knockouts. I think that's a great suggestion to really think about how what we can learn from human knockouts in terms of functional genes, but also in terms of future drug discovery and so on. And so the way we organize the workshop today is really to have some talks in the morning with some time for discussion. I do need the speakers to keep to time if possible. And then after lunch, we'll break out into 3 groups, and the groups go away and discuss particular questions and come back and report to the whole audience later on today. And the idea from this is that we will have, hopefully, a publication which comes out of it, which can be into the peer-reviewed literature, with some suggestions of how to move the field forward in that area. So we are recording today, and the reason for recording is that it goes on our website, and the most popular part of our website are the recorded talks, and particularly from patients actually. Our top hit is a patient-related talk, where they talked about a particular condition that they had, and so on. But these talks are then accessed by lots of people, and our website traffic has been increasing year on year. So I think it would be good that we do that, and we have increasing numbers of people now joining the network from outside the UK. Although it's called UK Network, we have a lot of people joining from outside the UK. So it's good that we disseminate as widely as possible, if that's okay. So I don't want to continue giving an introduction. I'm going to go straight to the first speaker. And that is Dr. Anna Need, who doesn't need any introduction, works in Genomics England, and Anna is going to tell us about opportunities for human knockout research using the data sets which are available in Genomics England. Thanks very much, Anna.
Thank you for the introduction, and I'm very happy to be here. I've been very excited about human knockouts ever since the East London Genes Project started, and of course we've got some fantastic opportunities in the 100,000 Genomes Project. So the 100,000th genome was collected in December last year. We've had continued very strong support from the government for genomic medicine and genomic healthcare. In October 2018, last year, Secretary of State for Health and Social Care Matt Hancock committed to sequencing, or government funding to sequence another 1 million genomes, which is half from the NHS, which will include all children seriously ill with a suspected genetic disorder and adults with select rare diseases and difficult to treat cancers. So that's 500,000, and then also 500,000 from the biobank, which will be good for basic research, but for also feeding into things like polygenic risk scoring to be integrated into the NHS. In addition to that, there is a lot of interest and commitment into sequencing more, potentially millions more genomes, and we're working with the government and other industrial and other funders in order to make a plan for that. So to date, we actually over-collected, so we've got 123,000 samples. This isn't 123,000 people, because for rare disease, to make it easier to identify the pathogenic variants, we collected trios wherever we could, and where we couldn't, we collected other affected and unaffected family members. And for cancer, we sequenced genomes from the tumour and from the germline, so we could look for differences that could be contributing. So far, we've sequenced 116,000 of those genomes. And you can see the breakdown here between rare disease and cancer. And we have returned results to the NHS for nearly 100,000, so we're getting close now. So when the genomes come to us, we do some initial, we do an analysis for the genes that we think are most likely to be causing that condition, and feed that result back to the NHS. And then the GMCs go through these results and see which of those they think are most likely to be causal. At the moment, it depends very much on the condition. There's different rates between different conditions, but on average, we have between 1 in 4 and 1 in 5 actionable findings for rare disease, and about half of cancer cases have something relevant fed back to them from the project. So what do we tell the participants? Of course, we tell them about things that we think are causing their main condition, so the condition for which they were originally referred. But also they have the option to get information about additional findings, so things that are irrelevant probably to their original condition, but they're serious and actionable conditions, for example, breast cancer variants, and also for carrier status for affected parents with rare disease, so you can see their likelihood of having other affected children. Everything that we do, so all the analysis that we do in the 100,000 Genomes Project is based on PanelApp. So PanelApp is a database of gene panels that's crowdsourced. So experts from all over the world have fed into PanelApp, and they have said that for particular conditions or phenotypes, which genes they consider to be most relevant. And then these panels are curated by experts within Genomics England, and the associations are colour-coded, so red if they don't have enough evidence, amber, and then green if they're considered to be very strongly associated with the condition. And it forms the basis of the results that we feed back to the NHS. So the doctors look at the patients and say, well, we think these panels should be applied. So the panel, for example, for skull abnormalities or intellectual disability. And then all the green genes in that panel are any pathogenic looking variant, any damaging looking variant in a green gene in that panel is flagged to the genomic medicine centres, and they look through all of those ones. They can also look through other ones, but they have to look through those ones. So this set of genes includes knockouts, and it includes also other types of variants, of course, that are gain of function, for example. And we also had a quick look last night to see how many would have to be that are recessive inheritance, so you would potentially need both copies knocked out. And there's 2,200 genes that are green in PanelApp that you need to have both copies knocked out before you get the disease. This also includes, so this is a brilliant database of the knockouts that we know cause disease in humans, but also other phenotypes, for example, drug response. I probably don't need to give any examples of pharmacogenomic variants to this room, but just as an example, I think a lot of the knockout work is focused on drug response because, of course, those types of variants aren't under the same selection pressures that knockouts that cause phenotypes per se are under. So you've got a lot of them that are very, very damaging with a big impact, but are at the same time common in the population. And one that we have started reporting back on this year is germline DPYD variants, which affect the metabolism of common drugs used in chemotherapy. And if you have both copies of this gene knocked out, I think you have phenotypes from a very young age, and it's recognizable. But if you have one copy of this gene knocked out, you don't tend to manifest any phenotypes. But if you're treated with one of these types of cancer treatment drugs, you are much more likely to have a toxic reaction, which can even be fatal. So by feeding these back, it's potentially going to have a very strong clinical impact. So we've got, as I said, we've got genes that we know are associated with disease. We've got genes that we know are associated with drug response. But we all know that there's so much out there that we don't know. There are genes that are associated with knockouts that are associated with disease, cause disease. There's knockouts that cause phenotypes that aren't necessarily disease, but are very important for us to understand, like how we metabolize food, for example, how likely we are to become obese, to store fat, which are relevant to disease. There are variants that are relevant to drug response that we don't know about. And there are variants that are important because they may lead to the development of new drugs. And many of them are very, very rare. So we know that we have to look at very large databases in order to find anybody with these variants. So we are very lucky with the 100,000 Genomes Project that everybody consented to the use of their genomes and their data for further research. And we've got two sets of collaborating partners that work with us on the research side. And these are the Genomics England Clinical Interpretation Partnership, which is the GeCIP, which I lead, and the Discovery Forum, which is the industrial partners that work with the genomes. And they do two types of work with the research data. One is to improve the rates of diagnosis. So by focusing on green genes in PanelApp and by variants that are very, very clearly pathogenic, we're sure that we're not reporting back rubbish to the NHS that aren't really going to be causing illness. And that's really, really important. But of course, we sequence genomes. We didn't sequence exomes or panels. We sequence genomes for a reason, and that's because there's so much still there to be found. And as you can see, that we're finding reportable findings between 1 in 4 and 1 in 5 cases. And in some cases, that's because there isn't a Mendelian variant to be found, but in many cases, that's because we don't yet know that the gene that the person's got a mutation in is associated with that phenotype, or they've got a splice variant that we can't recognize because it's in a part of the intron that we don't understand is relevant, or all sorts of reasons that make it worthwhile to do genomic research. So these partners, the Discovery Forum and the GeCIP, are doing basic research on the genomes, and that might be population genetics and things like that that will help us eventually in the longer term develop a better genomic medicine service. But also they're looking through the genes and improving the rates of diagnosis by, for example, just looking at genes that they know really well because they've investigated them for years in mice, or they're applying machine learning techniques to identify particular classes of variant that they can then feed back and work with the doctors to identify the most pathogenic ones of those. So the GeCIP currently comprises about 3,500 researchers worldwide, and they have they can get access to all of the data in the research environment. They are self-organized and they're self-funded, so they write grants. They're just normal academic or clinical researchers. There's a lot of enthusiasm in big cancer funding bodies in the MRC and Wellcome, amongst others, for work on the 100,000 Genomes Project. And you can see that there's a very good rate of return. So about half of the money that's applied for in grants that we support is awarded, which is pretty good. And there's £22.6 million worth of grants working on the project data at the moment. The GeCIP are divided into 3 areas. So rare disease and cancer are disease-focused, and you may get people looking for knockouts, for example, that affect cancer, cancer treatment, or individual rare diseases. If you are like screening across all of the genomes for knockouts, for example, or for methods to identify non-coding variants that are pathogenic, you would potentially be in one of the cross-cutting groups that work across the genomes, regardless of disease. The Discovery Forum have access to the same data and can do exactly the same research if they want to. It's usually a little bit more applied or translational, but they have helped us already to identify diagnoses in other participants. So what data do we have in our research environment? That's the sort of exciting part of this, and this is how we're going to start doing knockout research with the data. So we've got the genomes in there. We've got over 100,000 genomes in there now. And we've got the raw BAM files, so if you need to do new alignment for particular types of variant hunting, you can do that. But we've also got VCF files that we've called, we've got Illumina VCF files, we've got Illumina CNV file VCFs. So we've got a lot of data. Raw data. We've also got data sets that we have prepared, so that's including the tiering data, so the ones that where we've applied the green panels and pulled out the genes that we've fed back to the GMCs. That includes tier 3 data, so damaging looking variants that aren't in green panel app genes. We've got the feedback from the GMCs where they've looked through and said, oh yeah, we think that one is causal. And we can say, right, that case is solved. So you could, if you wanted to focus on unsolved cases, you could use those data. We've also got some fantastic data sets prepared by Lucas's team, who's here in the audience. And that includes aggregate files. They're working on de novo calls and all sorts of ways of bringing the data together. We've recently, in the last release, done some methods for calling ethnicity and ancestry from the genomes, and that's all in there as well. In addition to that, we've got the HPO terms that were put into the database by the doctors when they recruited the patients, so you can see the clinical terms that are relevant to their conditions. And we've also got secondary data that we get from NHS Digital, which includes hospital episode statistics, imaging data sets, mental health data, and other data sets related to cancer treatment and outcomes. So that's really important because it won't necessarily only be related to their recruiting conditions. So if you find an interesting knockout, you can go back and look at those data sets to try to confirm the phenotype. Oh dear. Oh yeah. So also all the data are kept in a research environment, which is a virtual desktop. We can't take the raw data out. You can take summary level data out, but you go into the research environment and you use the tools and the compute that we provide to do the analysis, which means actually that it's really available to anybody. You don't need to have your own compute system and all these tools loaded on your own computer. You can be a person starting up a research group. You can be a PhD student with an interest that's potentially different from your research group. And you can go in and you can carry out analyses on our high-performance compute cluster using our data with all of our tools and investigate your hypotheses. And that's because we've got central support from the MRC and Wellcome and others for GeCIP members to carry out these analyses. And we've got shared folders. We've just upgraded our storage, so it's amongst the top 10 in the world. I think they measure it every night. Sometimes we get to number 2, so it's very good. There are quirks to the research environment, but it's getting better all the time, and we're adding new tools and new ways to look at the data. This is something that is currently being improved all the time, but has really opened up the data to people who don't necessarily know how to run things on the command line on the HPC. So this is a tool that means that you can look up particular genes or variants and see what participants we've got with those particular types of mutations. I should have shown you a picture of the drop-down menus, but you can, for instance, say, show me all the genes that are green in the panel for intellectual disability, or show me variants in this particular gene, show me ones that are less than 1 in 10,000 in NOMAD, and that lead to loss of function in the gene, and or missense that are deleterious. You can do all of these filters, and then you can find the participants with those particular variants, and then link back to the phenotype data. So that's a very good way of looking for knockouts in our data set. So yesterday I took this paper and I looked, so many of you will know this paper better than I. This was an investigation of knockout variants in people who have got related parents, so obviously have got higher rates of homozygous loss of function. And I looked at a few of the genes that they picked out that had loss of function mutations that were associated with particular phenotypes. And I looked at this gene, which is associated with lower LPPLA2 enzymatic activity. But interesting, when they looked at heart disease risk, although they would have predicted that there'd be an association with that, there wasn't. But I looked in our database. It was, I think there were, I can't remember how many there were in here, but not many, maybe 1, 2, something like that in this database. And so you can see the results here. I can't actually from where I'm standing. We've got 32 people with heterozygous loss of function variants in this gene. This is all of the data. These are parents and unaffected parents. And we've got 3 complete knockouts for this gene. And we can go back and look at what phenotypes are associated or carried in those people. And you've got information about the variant at the top, and that's the same variant that was reported in the paper. What's really valuable about our data set for looking at human knockouts is the clinical data that's available there, and it's really detailed clinical data, and that's because you go into the research environment and you're not taking the raw data out. So I think it's one of the most detailed databases that we've got in terms of phenotypes. We've got family-based study design, so you can see if it's a de novo variant, or if it's inherited, and we've got the data on the parents as well, not just the affected child. So if it's a knockout that's irrelevant to their recruited condition, for example, it doesn't matter. You're still going to have the data for the parents as well if they've been sequenced. So all the people in the study are participants. And what is really exciting, and also still a work in progress, is recall for further phenotyping. So of course, it's great if it's related to their seizure disorder that you're trying to find knockouts. But if you're, for example, working on energy metabolism and things like that in rats, and you want to see whether the phenotypes are replicated in mice, or memory phenotypes or something like that, and you find in our database that there's 3 people with a knockout of your gene, you probably won't have that data in their clinical records, however rich they are. But what we can do is, and what the participants are very keen on doing, is working with us to provide extra phenotype data for further research. So we have got a fantastic participant panel. They're really engaged. We really don't make any big decisions without them. So, for example, on recall, they're very keen to do all of these new exciting things, like have apps on their phone where researchers can contact them directly and ask them questions. They're really very enthusiastic and excited. I'm not saying that we'd be able to do it tomorrow because, of course, there's always things to set up. But that is something that we expect our data set to be able to provide. So really it's, I think, one of the most fantastic data sets for finding knockouts and following them through and really following up hypotheses. So this is just a picture of some things that we've got now. We've got secondary data, genome data, biobank data is being linked in. And then some hopes for the future, like wearables, devices that many of the participants are very excited to wear wearable devices and provide real-time information on certain things. So we're a good way into this, but the future is bright. And just a quick plug for our research conference on the 4th of November. We've got several hundred GeCIP members coming and Discovery Forum members coming to present their research and look at each other's research in Central Hall, Westminster, and you can still register for the meeting now. Thank you very much.